Antisense oligonucleotide functionality and targets, Gapmers, Steric Blockers, Splice Modulators. Antisense oligonucleotides, ASOs, have emerged as a versatile and powerful tool in molecular biology and therapeutics, offering precise modulation of gene expression through various mechanisms. These short synthetic strands of nucleic acids are designed to bind specifically to RNA transcripts, leading to diverse outcomes such as mRNA degradation, splicing alteration, translation inhibition, and more. This article delves into the detailed mechanisms and applications of various types of ASOs, including Gapmers, Steric Blockers, Splice Modulators, miRNA inhibitors, siRNA, AntimiRs, and Exon Skipping Oligonucleotides. Gapmers utilize a central DNA region flanked by chemically modified RNA nucleotides to induce RNase H-mediated cleavage of target RNA, offering a potent approach for gene silencing. Steric blockers interfere with the binding of proteins or other molecules to RNA, modulating processes like translation and splicing without degrading the RNA itself. Splice modulators alter pre-mRNA splicing patterns to include or exclude specific exons, addressing genetic mutations at the RNA level. miRNA inhibitors, or AntimiRs, prevent miRNAs from interacting with their target mRNAs, thus de-repressing gene expression. siRNAs harness the RNA interference, RNAi, pathway to guide the degradation of specific mRNAs, providing a robust method for gene knockdown. AntimiRs, on the other hand, specifically inhibit miRNAs to modulate their regulatory functions in gene expression. Exon skipping oligonucleotides are particularly notable for their application in genetic disorders. By binding to pre-mRNA and blocking splicing machinery access to specific exons, these oligonucleotides can restore the reading frame of mutated genes, producing functional proteins and ameliorating disease symptoms. This comprehensive exploration of ASO mechanisms underscores their significant potential in both research and therapeutic contexts, highlighting their ability to precisely manipulate genetic and epigenetic landscapes for disease treatment and biological discovery. Gapmers, mechanism and structure. Gapmers are synthetic oligonucleotides designed to induce RNA degradation. They consist of a central region of DNA nucleotides flanked by chemically modified RNA nucleotides. The central DNA region forms a DNA-RNA hybrid when bound to the target RNA, which is recognized by RNase H, an enzyme that degrades the RNA strand of the hybrid. The chemically modified RNA nucleotides at the ends enhance the binding affinity and stability of the Gapmer, protecting it from degradation by nucleases and improving its binding specificity. Applications. Gapmers are primarily used for gene silencing in therapeutic contexts. By degrading target RNA, they can reduce the expression of harmful genes. This technology has been applied in treatments for neurodegenerative diseases, cardiovascular diseases, and certain cancers. Steric blockers. Mechanism and structure. Steric blockers are oligonucleotides that bind to specific sequences of RNA and physically obstruct interactions with other molecules, such as proteins or ribosomes. Unlike Gapmers, steric blockers do not induce RNA degradation. Instead, they prevent processes like translation or splicing by hindering the access of necessary molecules to the RNA. Applications. Steric blockers are used in therapies for Duchenne muscular dystrophy, DMD, and spinal muscular atrophy, SMA. They're also employed in research to study gene function and regulatory mechanisms by blocking specific RNA interactions. Splice modulators, mechanism and structure. Splice modulators are oligonucleotides that bind to pre-mRNA and alter its splicing patterns. They can block splicing enhancers or silencers, or redirect the splicing machinery to include or exclude specific exons. By modifying the splicing process, they can influence the production of different protein isoforms. Applications. Splice modulators are notably used in treating DMD and SMA. By altering splicing patterns, they can promote the production of functional proteins, which is critical in these genetic disorders. miRNA inhibitors, AntimiRs. Mechanism and structure. miRNA inhibitors, also known as AntimiRs, are synthetic oligonucleotides designed to bind to microRNAs, miRNAs. By binding to miRNAs, they prevent these small regulatory RNAs from interacting with their target mRNAs, thereby modulating gene expression. Applications. miRNA inhibitors are used in the treatment of cancer, cardiovascular diseases, and neurological disorders. By inhibiting specific miRNAs involved in these conditions, they can alter the expression of target genes to achieve therapeutic effects. siRNA, short interfering RNA. Mechanism and structure. siRNAs are double-stranded RNA molecules that guide the RNA-induced silencing complex RISC to degrade target mRNA. The siRNA binds to the RISC, which then uses one strand of the siRNA to locate and cleave complementary mRNA, leading to its degradation and subsequent gene silencing. Applications. siRNAs are widely used in research to study gene function by silencing specific genes. They are also being developed as therapeutics for targeting viral infections, cancer, and genetic disorders by degrading harmful mRNA. AntimiRs. Mechanism and structure. AntimiRs are oligonucleotides that inhibit mRNAs by binding to them and preventing their interaction with target mRNAs. This inhibition modulates gene expression by blocking the regulatory effects of miRNAs. Applications. AntimiRs are used in the treatment of cancer, cardiovascular diseases, and neurological disorders. By modulating miRNA function, they can influence the expression of genes involved in these conditions. Exon skipping oligonucleotides. Mechanism and structure. Exon skipping oligonucleotides are designed to bind to specific exons in pre-mRNA and induce their exclusion from the mature mRNA. This technique can restore the reading frame of mutated genes, leading to the production of functional proteins. Applications. Exon skipping is used to treat genetic disorders such as DMD. By excluding specific exons, these oligonucleotides can produce functional proteins despite the presence of genetic mutations. Biochemical modifications in Antisense oligonucleotides. Antisense oligonucleotides, ASOs, are powerful tools for modulating gene expression. Their efficacy and stability are significantly enhanced by various biochemical modifications. Here are the primary types of modifications used in ASOs: phosphorothioate, PS, backbone modification. Description. In this modification, a non-bridging oxygen in the phosphate backbone of the oligonucleotide is replaced with a sulfur atom. Benefits: enhances nuclease resistance, improving the stability of the ASO in biological environments. It also increases binding affinity to proteins, facilitating cellular uptake, 2-O-methyl, 2-O-me, and 2-O-methoxyethyl, 2-MOE. Description. These modifications involve the addition of a methyl or methoxyethyl group to the 2-carbon of the ribose sugar. Benefits: improve RNA binding, affinity, and nucleus resistance. They also reduce immune stimulation, making the ASOs more suitable for therapeutic use. Locked nucleic acid, LNA. Description. LNAs have a methylene bridge connecting the 2-oxygen and 4-carbon of the ribose. Locking the ribose in the 3' conformation. Benefits: greatly increases binding affinity to complementary RNA sequences and enhances nuclease resistance, making LNAs highly potent. Peptide nucleic acid, PNA. Description: in PNAs, the sugar phosphate backbone is replaced by a peptide-like backbone. Benefits: PNAs exhibit high binding specificity and affinity to DNA and RNA. They are also resistant to enzymatic degradation. Phosphoramidate morpholino oligomers, PMO. Description: PMOs have a backbone consisting of morpholane rings connected by phosphoramidate linkages instead of the traditional ribose and phosphate backbone. Benefits: highly stable and resistant to nucleases and proteases. They do not induce immune responses, making them ideal for the therapeutic applications. 2-fluoro-2F. Description: this modification involves replacing the hydrogen atom at the 2' position of the ribose with a fluorane atom. Benefits: enhances binding affinity to RNA and increases nuclease resistance, improving the stability and efficacy of the ASO. N-acetylgalactosamine, GalNAc. Conjugation. Description: GalNAc is conjugated to ASOs to target hepatocytes specifically via the Asia-log-glycoprotein receptor. Benefits: facilitates targeted delivery to the liver, enhancing the therapeutic index of ASOs used to treat liver-related diseases. These biochemical modifications are crucial in optimizing antisense oligonucleotides for therapeutic use. By enhancing stability, binding affinity, and specificity, and by reducing immune responses, these modifications enable ASOs to effectively modulate gene expression for the treatment of various diseases. Conclusion: antisense oligonucleotides, ASOs, represent a transformative class of therapeutic agents with the capacity to precisely modulate gene expression through a variety of sophisticated mechanisms. The diverse types of ASOs, including Gapmers, Steric Blockers, Splice Modulators, miRNA inhibitors, siRNA, AntimiRs, and Exon Skipping Oligonucleotides, each offer unique modes of action and therapeutic potential. Gapmers stand out for their ability to induce targeted RNA degradation via RNase H, providing potent gene silencing capabilities. Steric blockers offer a non-degradative approach, disrupting critical RNA-protein interactions to modulate gene function. Splice modulators manipulate pre-mRNA splicing, allowing for the correction of genetic defects at the RNA level. miRNA inhibitors and AntimiRs effectively neutralize miRNAs, lifting their repressive effects on mRNA targets and altering gene expression profiles. siRNAs harness the RNA interference pathway to achieve precise gene knockdown, proving invaluable in both research and therapeutic settings. Lastly, Exon Skipping Oligonucleotides enable the production of functional proteins by bypassing defective exons, offering hope for the treatment of various genetic disorders. The advent of these ASOs has revolutionized our approach to understanding and treating genetic diseases, providing tools that can address conditions at their molecular roots. Their applications span from basic research, where they elucidate gene function and regulatory networks, to clinical therapies that tackle previously intractable diseases. However, challenges such as efficient delivery, off-target effects, and immune responses remain and are the focus of ongoing research and development. As our understanding of molecular biology deepens and technologies advance, the scope and efficacy of antisense oligonucleotide therapies are poised to expand further. Continued innovation in this field promises to bring forth new treatments and potentially curative interventions for a wide range of genetic conditions, underscoring the profound impact of ASOs on the future of medicine.